So let's see. People are coming in. This is pretty neat how it just rolls down, you know, it just, people just keep logging in. It's kind of cool. I see some new names, which is nice. So we're about 1 minute away from starting. And so what we'll do is kind of what we discussed. We'll turn it over to Damien real quick, then I'll get Lee done, and Lee will just let you do whatever you want.
Okay, sounds good.
And you want to take questions at the end or in the chat room?
I'm going to take questions a little bit at a time because this is a complicated topic. I've really worked hard to make it simple, but I do want to catch people and not have them like give up, you know. So okay. I'm stopped. There's like 4 times that I'm stopping and saying, okay, let's see if there are questions. And then I've saved quite a bit of time at the end for questions.
Okay.
And then if there's no questions, I have backup slides. So, you know, we're good. We can fill the time.
We'll talk about the Martinez's. How's that?
Right. Oh, yeah. And for the next 3 hours.
Yeah.
Yeah.
The never ending name of Martinez's.
Yep.
Okay. So I hope everyone can hear me and we're all settled in this morning. My name is Henrietta Martinez Christmas, and I'll be hosting the lecture today by Dr. Lee Martinez. But first of all, I'm going to turn it over to Damien Aragon, president of the New Mexico Genealogical Society.
Thanks, Henrietta. Yes, I'm Damien Aragon. I want to thank everyone for joining us for this talk by Lee Martinez. But before he speaks, I want to remind everyone of the upcoming annual conference. It will be a one-day conference on Saturday, the 15th of October. We will have 4 speakers that day, plus the added bonus of 8 more on-demand talks. All of the talks will be available until November the 15th. Also, we will be announcing the prize winners at the end of the conference. So go to nmgs.org, register for the conference on October the 15th. I'm going to hand it back to Henrietta.
Thanks. Yeah, I hope to see everyone at the conference. So today we are so happy to have Lee Martinez speak to us. He spoke at one of our conferences, and I keep wanting to say it was like 2018, but you can't quite remember. For those of you that want to get more familiar with Lee, he has quite a YouTube channel following, so you can just Google Lee Martinez, DNA, YouTube, and he's got a number of talks. You can go listen to him speak on various other DNA topics. He is a native New Mexican, and he's got that great name, Martinez, like I do. Just love that. And so Lee, you want to get your slides up, and I'm going to disappear from here, and so is Damien, and we will turn it over to you just as soon as you're up. I can't hear you.
All right. Welcome, everybody. Thanks for having me. I look forward to this presentation. I'm so glad that you're here. So I have multiple screens. So if you see me doing one of these things, that's what we will be up to. Today, I want to talk about genetic genealogy. There's so much going on right now. I mean, it's moving so fast. and it's hard to keep up with it. And I wanted to kind of bring everyone up to speed. It's moving so quickly that some of the tools I'll be showing you today, I actually hadn't seen until this morning because they just came out Thursday. So that's how quickly, on a weekly basis, almost, there's new tools and new things coming out. All right. All right. So today I'll be introducing two new tools, most likely to be referenced in a scholarly journal, like the New Mexico Genealogist, on the topic of genetic genealogy. The Big Y block tree and the scientific details section of the new Discover More tool, and we'll explain what those are later. I will also introduce the newest Discovery More features. Like I mentioned, some of them just days old. So the goal of this presentation is to prepare you to be able to read, understand, and interpret genetic genealogy findings you may find in the New Mexico Genealogist moving forward, and leverage some of the newly available tools. Now, because there's a lot going on here, there will be pauses throughout the presentation to respond to questions. I may, as I hear the question, I may put it in the parking lot because to talk about towards the end. If it's something that we're kind of in the middle of talking about, I will address it at that time. But I want to make sure that we bring everybody along. We probably have folks here that are like really expert in this and are on top of it. Others, maybe you don't even know what Y-DNA is, and this is your first time checking it out. So I want to be thoughtful about the audience and make sure that I don't go too far, too fast. So like lots of opportunities to and time for questions. All right, so I want to start off talking about starting off basic with one of the fundamentals of genetic genealogy, which there are two trees. We have our family tree there on the left that genealogists use to document the results of their research with ancestors, descendants, and life events like marriages, births, and deaths. And then we have the genetic tree there on the right, and it's built based on genetic markers from hundreds of thousands of DNA test results. And these trees are constructed entirely without any historical records or genealogies. They span all known paternal and maternal lineages in the world over hundreds of thousands of years and connect every person on the planet. And genetic genealogists leverage both trees. Y-DNA testing can help you discover and verify your direct paternal ancestry by connecting you with other individuals who are descendants of a shared common patrilineal ancestor. Now, some people often think, well, wait a minute, that's just one line. And, you know, I'm a genealogist, I have hundreds of lines. Well, I would say that, well, if you have hundreds of lines, half of those lines are Y-DNA lines. So just think about that. This Y-DNA covers 50% of all of your genealogy. I want to focus on the connection. I want to encourage you to willingly share your data as widely as possible. So this is fundamental of any type of genealogical research. And I say this in behalf of those who have been working in this field and who reach out to DNA matches and then they don't hear anything back. So I just want to encourage folks to engage in this effort together. Another basic thing I want to mention regarding Y-DNA is that there are two kinds of Y-DNA mutations, really two different tests. One is the short tandem repeat based STIR based tests, the Y37, the Y67, the Y111. Think of that as like a stutter in the on a line of code. So here, as an example, we have this CTA, 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 and it repeats 5 times. So let's say that we do a Y37 test. So you have, and this is just one of those 37 markers. We're just looking at one of those 37 markers across 3 people that are matches. We could say that these 2 folks are 1 genetic distance apart. And then these 2 folks are 2 genetic distance apart. And there's a whole science about genetic distance at 37, genetic distance at 111. We're not going to go into that today because, quite frankly, STIR tests are kind of old, and we really don't use STIR tests anymore as the primary method of doing Y-DNA research. And this is why I'm talking about the movement in Y-DNA. It's moving so fast. So as a comparison, mitochondrial DNA, Mitochondrial DNA test today is exactly the same test as it was 10 years ago. No difference whatsoever. The Y-DNA test, it changes almost every year. We had the Y12, and then it was the Y25, and the Y37, it became the Y111, then it became the Y500, then it was the Y700, which connects with the big Y700, and then just mark my words, another couple of years we'll have something else. It's moving so quickly, and the focus of the tools, which is the focus of today's presentation, all the tools that are being built are all being built for these SNP-based type of mutations, like the big Y700. So we're going to be talking about these kind of mutations today. It's a single, and we're going to go, trust me, you're going to understand this really well by the end of the presentation today. It's a single change in a marker. So in this case, this person had a mutation at this location, and this creates what's called a branching point. And we'll show that here in a second. But branching points are really important as we use and leverage the data to be able to do genealogy in genealogical time. That's another thing I want to mention, is that a few years ago, STIR testing was being used instead of SNP testing, because SNP testing was seen as something that was old and ancient. That's outside of genealogical time. Neanderthals 10,000 years ago, 600 years ago, and STIR testing was seen as, oh, this is stuff within genealogical time. Well, that's no longer true. SNP testing is now gotten to the point where it comes all the way up to the present time, and so it has overshadowed STIR testing. All right, so today I'll be introducing the big Y block tree, because this is something you're going to see, I would expect, in the next year to 2 years, in any kind of DNA related article, a Y-DNA related article in the journal. You're going to see, I would expect at some point, either this picture like this or a reference to it. So I wanted to kind of introduce you to it so you understand what's being reported. It's a display of the big Y tree structure with a SNP scale. So these are all SNPs. These are those mutations that I was showing you, right? Those SNP mutations. Think about it as a family tree of SNP mutations. And they occur on average, and there's a lot of variation in that, of every third generation or so. The block tree highlights how many genetic mutations have been associated with each lineage on the tree. With a rough estimate of the time that typically passes between mutations, one can count the number of SNP mutations on a lineage and make inferences about when a specific shared ancestor may have lived. Now, interestingly enough, what I just said is actually old news. 5 months ago, it would have been like the latest and greatest. What we would do is we would count these mutations, and then we would multiply by 84 years, because on average, it's 84 years per mutation. Then we would come up with an estimated date. Well, now, Family Tree DNA has created a new algorithm where it uses not just this information, but all that STIR data that we said we don't use anymore. They've created a way to grab all that data and all this data and come up with a more refined time to most recent common ancestor. That is huge, because we want to know, when did this happen? Because if you have a genealogy that's well developed, you can lay your genealogy right on top of this block tree and start to put names to these mutations. And then if you share a mutation with this other person, that other person doesn't have any genealogy, you know that your genealogy is going to connect at a certain point in time. So this is huge, and I want to show that today. All right, so every big Y block tree is available to anyone who has taken the big Y700 test via the block tree app. It is not only allows you to see your own big Y block tree, but anyone else's as well. So if you've taken the big Y test and you go in and you want to see someone else's big Y, all you have to do is go in the upper right-hand corner and enter in the haplogroup, if you happen to know what their haplogroup is. And you'll, again, refer to those journal articles. The journal articles will reference haplogroups. And if you're interested in learning more about those haplogroups from those journal articles, you just enter it in here and it will bring up the block tree. And it's totally shareable, because so if it was my test, I would not just see this block tree, but I would see all the names of the testers. all my matches. You wouldn't see that because that's not, that's kind of personal information, right? So that's kind of cool, is that you can go and see anyone else's branch and not see their personal information. So it allows people to share and collaborate without worrying about sharing personal information. This navigation function allows you to see all the information on any given block tree with the exception of test takers' names and matches. And this view allows researchers the ability to share and publish. Okay. All right. The analysis of your Big Y test results are presented in the Big Y block tree, which is available to customers, but it's also available on the haplotree, which is available to the public. So even if you haven't taken the Big Y test and you're reading an article and you're interested in learning or seeing more information about that, you can always go to this website right here and type in the haplogroup, and you will see this haplotree, which is the same thing as the Big Y block tree minus all the mutation history. So if I have this family clade QFT5172, it's right here. And it's a subclade of this, which is here, which is a subclade of this, which is here, and so forth and so on. So it gives you that tree structure. It's available. And I want to make sure I'm using correctly. So here again, let me do that again. QFT5172 is here, and that's right here. It's a subclade of this, which is here, which is a subclade of this, which is here. See that? So as you go up, you get that same structure, that same information. You even get the number of mutations that are within each one. What you don't get here is the actual list of mutations, which you probably wouldn't be able to, that wouldn't really give you any information anyway, unless it's your line. And we'll go into that a little bit more detail as well. So my point is this: even if you haven't purchased any of these products, you can, and you're reading about it in a journal, you want to learn more, there are websites to go to see information and dig into it a little bit more. All right. FTDNA. I know, by the way, I have no affiliation with the FTDNA. I'm not, associated with them in any way. I don't get like compensated for like helping them sell stuff or anything like that. So I just want to throw that out there. I'm just a super user of their products and like to share the stuff I know about what they put out. So they provide a historical and prehistoric genetic family tree with a time perspective called the time tree. And this time tree has been out for only a few days. This actually got published on Thursday. So we'll be looking at it together a little bit later for the first time. But this is what I'm talking about. It's moving so quickly. The tools are developing so fast. They're literally pushing things out weekly now. So we'll look at that, and it's kind of cool. All right, I want to show you my home page, and there's nothing here that is proprietary. So I'm going to go ahead and bring it up here. The only thing on here that might be personal is my kit number, and I give permission to share my kit number, so don't worry about it. I share my kit. If you don't know my kit number already by now, well, now you do. I put this out readily, so I don't care. So this is my information. I'm going to switch screens over here so I can look at my, so I can just show you kind of where things are located. So remember I mentioned that the STIR tests, those STR tests and SNP tests are two different tests. It seems like when you take a Big Y, it's an upgrade from the lower level tests, the Y37, the Y111, but it's not an upgrade, even though it looks like an upgrade. It's a different test. They have to redo the test from scratch. So even though it looks like an upgrade from the purchaser's point of view, it's not. It's two different tests, two different mutations are looking at, two different pieces of science. So you have here, if you took those lower level tests like a Y111, which is really entry level now, it used to be the Y37 in the last several years. This is kind of like your toolbox. And if you take the Big Y, then you kind of get a new toolbox. And then there's this Discover More, which is brand new, which we're going to look at today. A lot of the stuff you're going to see in journal articles moving forward are tools that are in this Discover More toolbox right here. Here's my autosomal DNA. So, and then here's my mitochondrial DNA. And then here's my Y-DNA family clade and my mitochondrial DNA. And so anyway, so that's kind of how it looks. If you're in a project, so here you can see all the projects I'm on. So here's the New Mexico Genealogical Society DNA project right here. And lots of projects. I will point out that the projects have been around for a long time, right? They've been around longer than I have with Miguel Torres. But because they've been around for so long, they're actually based on old technology. They're based on they're stir marker based. So what's happening is that we're kind of transitioning from using this project platform, which is based on stir markers, to a SNP based project platform, which is the tools that you find in Discover More. So that's how I conceptualize it. Discover More is the more advanced version of the old projects that you may have been on. And of course, I'm in both, right? Because we're transitioning, and it may take a couple of years to kind of maybe move from here over to here. And I'm not even sure what that would look like. But most of the data in here we don't really use anymore directly, but it's being pulled and leveraged in Discover More. They're pulling the data. All right. All right, Family Tree DNA Discover, the Discover More tool, is also super accessible. Anybody can get to it, even if you've never purchased anything from them and have no interest in purchasing anything from them. You're reading a journal article and you see a haplogroup and you want to say, I want to leverage all these tools and views and I want to see more detail about what I saw in that journal article. You can get to it for free. You just go to discover.familytreedna.com. Right here, you can see the website, and you can go there now if you want and take a look. And if you have a haplogroup or you know of a haplogroup, you can enter it in and you can start looking at the tools now if you like. All right, so if you're interested in learning more about Y-DNA stir testing, such as the Y37 or the Y111, and how the projects use that kind of data or anything like that, as Henrietta mentioned, I have a ton of videos. So I would, here's the kind of the link to most of my YouTube videos. I'll actually see if I can find it here. Yeah, right here. There are lots of good YouTube videos on here. But these are really long. They're just like 4 hours worth of content. I will point out that this one right here has 4,000 views, which is even more than the famous Brent Cruz Francisco Montes Vigil presentation. So it's the #1 viewed DNA video that's out there for New Mexicans. So I just want to throw that out there really quick. If you want more information than I'm giving today, because I'm just blowing through the tools really fast, and you're like, slow it down, or I see that you're like moving beyond stir test, but I only have stir tests. I've only taken the Y37 or Y111. I have no interest in taking the big Y. I want to learn more about that. There's videos for that. So that said, I'm going to take a pause here and see if there are any questions before I move on and start jumping into the actual meat of the presentation. Okay, and we have several times where we're going to be able to stop for questions. So you're going to have lots of opportunities to ask questions. All right, so before I dive into the toolbox itself, I'd like to talk about the important role those folks play who are actively using these tools as genealogy citizen scientists. Often when we hear the term citizen scientist, we may think of amateur astronomy. That's what I think about first and foremost. Butterfly counts, or even art history. Citizen science is scientific research conducted in part or in whole by amateurs, and it's sometimes described as, quote, public participation in scientific research, close quote. Well-known citizen scientists include Isaac Newton, Florence Nightingale, Benjamin Franklin, and Charles Darwin. Less well-known researchers include folks like Judy Schmidt, who has been processing raw space data into breathtaking images for years. Recently, she has used data from the newly deployed James Webb Space Telescope to provide absolutely jaw-dropping images of 2 spectacular spiral galaxies. And this involves painstaking work. We should all be appreciative of the gift citizen scientists give of their time and the invaluable contributions they make to science. And these are the folks that you'll see writing these articles, right, and putting it into the journal. I want you to have an appreciation for all the work that they're putting into that. And And if you have opportunity and you see over the years these authors, reach out to them, thank them for their work. This applies to the relatively, of course, new field of DNA-based genealogy research. So we have folks like Miguel Torres, who's been a driving force, right, for many, many years, organizing y-DNA data into projects, like I pointed out earlier, for the New Mexico Genealogical Society. And recently we have Daria Celeste Landris, who has decided, I'm gonna go and drink the Kool-Aid. I'm gonna jump into this stuff. And so you're going to be seeing, I anticipate, some great articles from these folks and others. So if y-DNA research seems daunting to you, there are people out there who can help you within the Genealogical Society that you can reach out to. Over the last 25 years, and especially the last 10, commercially available DNA tests have revolutionized traditional genealogy methodology, enabling the amateur genealogist to participate as citizen scientists and expanding the family tree of mankind. And out of the various types of DNA tests available, advanced next generation sequencing, or NGS, y-DNA SNP testing using FTDNA's Big Y700 comes the closest to true citizen science. You're actually creating data and adding to the science in a way that you can't really do with any other kind of DNA test currently out there. Participating as a citizen scientist comes at a cost, however. It requires an educated group of skilled practitioners that are enabled to use the tools now at their disposal. So there is a learning curve, and it's exhausting because just when you think you've mastered it all, a new iteration of the test comes out, and or new tools come out, and then you have to still learn those. So you never can just sit down and say, okay, I've mastered it. It's a continually evolving thing. Some may not want to take the time to master these new tools, and others may find it difficult to accept the information that comes through them. But it is important that we learn and incorporate these skills into our practice and welcome the data as not only equal to, but in many cases superior to traditional genealogy records, which can be doctored or based on an incorrect understanding of the facts, as unfortunately Mr. Red discovered in this cartoon here. It's important to understand that y-DNA results may confirm or invalidate deeply held beliefs about your patrilineal line, and this can be hard. And those who have read or watched my videos know that I've had my own kind of moment, right? The last time I presented at the New Mexico Genealogical Society was the last live one that we had, which whatever year that was. So the one before COVID hit. And if you were there, you may recall that I talked about my 4 patrilineal lines, my paternal grandfather, my maternal grandfather, my grandmother's fathers, and of those 4, so Martinez, Merida Sanchez, Archuleta, Merida Trujillo, only the Trujillo line, y-DNA came back and said, oh, your Trujillo surname comes from the progenitor of the Trujillo line. All the other 3 lines, no. So yes, I'm a Martinez, but it turns out that I descend patrilineally from a Native American line. My Sanchez line, actually, the Sanchez surname wasn't around for that long before it becomes something else. The Archuleta line does not descend from Ascencio de Archuleta. It actually descends from a Pueblo Indian boy. And so, yeah, you just have to be ready for that. I had really good trees, really good documents, birth certificates, marriage, but the fact of the matter is that DNA can come in. It's going to trump your paper record. If you're saying I descend from Ascencio de Archuleta patrilineally, he was from the Basque area of the Iberian Peninsula. If you're in a QM3 haplogroup, that's a Native American, I'm sorry, but there's just no way that can be true. One of those has to be accurate, and the DNA will always be the one that's accurate. So you just have to get used to the emotional, you spend decades of time on a line, and I can tell you, I've had to come and literally shear whole portions of my tree off my tree because of y-DNA. At the same time, it's the ultimate validation of a line. There's absolutely 100% accuracy that this is truly your patrilineal line. So my recommendation is to not get too attached to surnames. Chances are high you will discover your surname has changed often over the centuries. So again, for me, we've been Martinez for about 130 years, which is a blink of an eye, right? I've been alive for 40% of that time. 130 years is really nothing. Before that, it was Martin for about 200 years, and then it was Espinosa before that, and then it was Gomez de Portugal before that, and then it was no surname because Kashkan tribal Indians didn't have surnames in the 1600s. So it's like, you know, over the time of 400 or 500 years, that's 4 or 5 surnames. They keep moving. So some advice there. On the other hand, in my Trujillo line, the Trujillo line just keeps going and going and going. So you'll have every once in a while a line that sure enough it matches to that progenitor, and it will keep going. All right, so I'm going to stop again, see if there are any questions before I proceed to the toolbox. And I'm going to look at the. I wasn't looking at the Q&A section, so let me just open that really quick. So I had a question, just making sure I'm tracking, is the Big Y the only one that does the SNP testing? From family tree DNA it is. There are other companies that do SNP testing, but if you want to do SNP testing with genealogy and you want to have your matches and you want to be able to contact your matches and do research, Big Y is really the only game in town currently. And of course, they have the biggest database. And that's really important. When people ask, which autosomal DNA test, Lee, should I take? You know, which is the best one? Is it My Heritage? Is Ancestry? My response is there's no real fundamental difference in the technology, in the science, in the chemistry in those tests. So the big difference is the database size. And Ancestry DNA is what I recommend. Why? It's not because I think they're better. It's just that their database is twice as big as the next biggest one. It's bigger than all the other databases put together. And so it really is about database size. And so Big Y, Family Tree DNA's Big Y has the platform to be able to share the information, and it has the biggest database by far. So I guess you could say Big Y is the ancestry for Y-DNA. Let's see, other questions. Are you familiar with Y-Seq? Yes. If so, have you found it useful resource in researching DNA? And would you have time to speak about it? I won't have time to speak about it. I have used it. I haven't really found a lot of value because Y-DNA, back to my original, this is what Family Tree DNA I think is trying to do. They're trying to put Y-Seq out of business. How do you do that? Make it so that there's no reason to go anywhere else. Bring in all the tools. So what happened is you had these specialty providers building tools that allowed you to do analysis and research. And so they would ask you sometimes to move your data over, things like that. And Family Tree DNA figured out, well, you know what? Why don't we just build these tools in-house? And that's why you're seeing right now this rapid building of tools on every Thursday. Every Thursday, a new tool has been coming out over the last 2 months. It's just all of a sudden out of nowhere, it's like bam, 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 bam. And I'm seeing they're duplicating these other organizations, and I really don't see a need for them personally. As long as I have organization putting it all into one place where I can do my work, there's no need for me to be using other platforms that don't really add value. But I will say that's my personal opinion. Next question. How do you pick who to test on the Big Y if you can't afford to test everyone? That is an awesome question. And because there is an answer for that, there's actually an entire set of videos on that. It's called the Rule of 4. I don't have time to go into the Rule of 4, but I want you to know that folks in the New Mexico Genealogical Society that are doing these articles that you're going to be seeing coming out and have seen coming out, they know how to do this. So when they reach out to you and ask you to test, it's not you're not being selected randomly. There's a reason. And anyway, Rule of 4, take a look at my videos. I really talk about this in depth, but there's not time to get into the detail. But there is a way, there is a methodology on being smart, because you can waste a test. Really quick, an example, let's say I took the test and my dad took the test and my brother took the test. Well, that's a waste. Me and my dad taking the test adds value, but then once we throw my brother in there, or you know, an uncle, then it's just too much. I would have been better off finding a fourth cousin, patrilineal cousin, to take the test. So there's a science behind it. Like I said, that's a great question, because you can't, it's not just a matter of just randomly picking as many people as possible. Hey, everyone in the all first, second, third cousins, uncles, nephews, take the test. No, you'll end up throwing a lot of money away if you did that. Can you speak about YFOL? They asked my husband to upload his DNA to that site, but I'm reluctant. Again, why upload the data? Anything that YFOL used to be able to offer, Family Tree DNA has basically built. And we're going to go into that today. You're going to see the tools that they're offering. So when you're in YFOL, ask yourself, is this being answered already? And the chances are probably yes. So that's all the questions so far. Great. Sorry, I had missed the Q&A section. So I like, there's no questions, but apparently there were. All right. Now let's get started. So this is my personal big Y block tree. It's a pedigree of SNP mutations which identify patrilineal ancestors. So this is the thing I want to point out, is the branching points. This is super important when you're looking at reading those journal articles. So I just want to point out, so here we have, you don't see the full block. These are called blocks of equivalence. Okay, blocks of equivalence. And you don't see the top block, but it is haplogroup BY66731. And it was estimated, the person who's representing this block was estimated to be born around 2,500 years ago, plus or minus 700 years. So let me stop there. How do I know that this person, well, let me back up. A block of equivalence, and this is kind of complicated, and now don't have a lot of time to dive into this. There's a video on it, but we don't know. It's a genealogy, right, of SNP mutations, but we don't know the order. We don't know which is the older than the next. We just don't know the order. And as we do research, what we do is we split the block. So there was a point in time when all of this was all when I first started, QL663 was the block. All of this was in this block. And what I've managed to do is split the blocks into smaller granular pieces, creating branching points. And that's what we do when we're selecting testers. But I know that so what I usually do is I usually say, and this is me, this last mutation, it represents the person who fathered these three. It doesn't mean that this mutation right here, it doesn't mean that this mutation right here was the one that fathered it. We don't know which of these mutations fathered these three. It's one of these, but we don't know which one. So I'll say this is going to stand in for now until I know more. And this mutation right here, I know that this person, because each one of these, and this is critical, each one of these mutations represents a human being. It's a person. If I had magical power or a genie or something, I say, give me the name, there would be a name that would show up here. Who is this person that fathered these three lines? Who is this person that followed these three lines? Who is this person that followed these two lines? I actually know who this person is that followed these two lines, because where I'm now in genealogical time and I have genealogy, I can lay on top of that. But the point I want to make is that what you get here is branching points, and the branching points, especially when you get down into genealogical time, these branching points become critical, because you're going to want to know, oh, I have this common ancestor. Which line do I come from through this common ancestor, or which son? So think about that. If you're working on a project and you have, you know, the Martin Serrano line, I'll actually show you that one later. Which of those sons do I descend from? As we add more data, we start to refine and create more and more branching points. And each one of those branching points is a place where a mutation happened and there was a split. If you're not following, we're going to spend a lot of time on this, so we'll get there. So again, this person here estimated to be born around 2,500 years ago, plus or minus 700 years. All right, so there's a range. Not everyone on here may know how to interpret statistics, but we'll go into that a little bit later as well. Just know that we have an estimate and a range of that estimate. So this one right here, this person I know was born around 800 years ago, plus or minus 250 years. And then this person here was estimated to be born around 300 years ago. And I actually know the exact date because, again, now I'm in genealogical time and I have the record, right? So again, this is totally shareable. Remember, you can look this up, anybody's haplogroup. If you see these at the very top here, these are the haplogroup names. And so when you see this, you just, if you're interested, you can go in and you can enter this into those toolboxes I'm showing you and find more information. All right. In order to understand how all of this works, we need to back up a bit. We need to kind of back away and start with some real fundamentals so that we understand what the heck this block tree is that we're looking at. So we're going to do that next. So in order to understand Y-DNA results, we need to learn some basic terminology from genetics and microbiology. Now, don't get freaked out. I promise to keep this super simple. There's nothing I will be presenting here more advanced than what you would learn in the first week of a biology class in 9th grade, okay? There are 6 concepts, really simple. Atom, molecule, nucleotide, that's a key one, DNA, chromosome, and genome. So we're going to go, each one kind of is a building block of the next. So we're going to go really quick and very simply into that. So we'll start with an atom. So this, the basic unit of matter. This is an oxygen atom. So it has protons and neutrons and electrons, and you don't need to know that for this. Just know that, all matter is built up out of atoms, right? And an oxygen atom is one we can think about. And then we have a molecule. A molecule is simply a group of atoms that are bonded together. So you have that oxygen atom I was talking about. And then if you put 2 hydrogen atoms to it, right, then you get H2O, or water, right? So this is a molecule. We all know this molecule because we deal with it every day. It's water, and it's very simple. It's made of 3 atoms, 2 different atoms with 3, H2O, right? Then all you need to do is throw in some nitrogen and carbon to that list of building blocks we've talked so far, and you have all the ingredients for the molecule we know well, which is caffeine. And so the only elementary building block that we're missing to make DNA is phosphorus. So this is a nucleotide, and this is a real key term for Y-DNA for people to understand. And it's easy. A nucleotide is a type of molecule, like that water molecule we saw, and is one of the 4 structural components or building blocks of DNA. So DNA is simply made up of 4 versions of a nucleotide, right, that you see there on the left. So the big Y700 tests for a change or a mutation or a polymorphism of a single nucleotide. So this is a nucleotide, this is a nucleotide, this is a nucleotide, and this is a nucleotide. So a single change in one of these, polymorphism change, single nucleotide polymorphism, or SNP. So it's just one of these letters changing, right? And that's what we're tracking in that big Y block tree. So these 4 structural components or nucleotides are strung together in long strands, forming a double helix. And this is even super simple because it's not random, right? It's the T always matches with an A, always. And likewise, an A always matches with a T. A C always matches to a G, and likewise, a G always matches to a C. So if I only knew half the information, I could easily reconstruct the other half because it follows a real basic set of rules. And in fact, that's how the cell knows that rule, and that's how it's able to make new strands of DNA and how you get new cells. These strands are paired up and wound into larger structures called chromosomes. Humans have 22 pairs of autosomal chromosomes and 1 pair of 6 chromosomes. So 23andMe, right? That's where that concept comes from, 23andMe. So of those 23 pairs, 22 autosomal chromosomes and 1 set of 6 chromosomes, if you end up with 2 X chromosomes, you're a female, and if you have an X and Y chromosome, you're a male. That's why only men can take the Y-DNA test. So let me stop right there. Like, well, if only men can take the Y-DNA test and I'm a woman, how does that help me? Well, if you have a brother, a father, an uncle, a first male patrilineal cousin, then you simply just recruit that person to take the test for you. And that's what I did for my paternal grandmother and my maternal grandmother, right? My paternal grandmother was a Sanchez, so I found a Sanchez nephew of hers. And my maternal grandmother was a Trujillo. I found a Trujillo nephew of hers, said, I need your DNA. And I'm good. So the fact that you're a woman doesn't keep you from leveraging Y-DNA testing at all. And if you're a male and you have female lines, you want to know their father's line, you just find you just recruit a nephew, a cousin, a brother, a son, and you're good. So the genome is the complete set of genetic information in a person. So the entire encyclopedia of instructions on how to make you. So that's it. That's all you need to know. Bonded atoms create molecules. A special set of molecules make up the 4 nucleotides. Those 4 nucleotides make up a structure called DNA. DNA are paired up and wound into larger structures called chromosomes, and all those 23 chromosomes make up the genome. That's it. So what does all this have to do with the big Y block tree? Well, everything. The big Y block tree is a pedigree of SNP mutations, those nucleotides, right? Every line you see here represents a single nucleotide polymorphism, a variant, a mutation that happened to a specific person in utero that was then passed along to all their male descendants for all time. And these variants occur on average, like I said, every 3rd generation or so. So it creates like a metronome or a time clock that allows us to start building estimates of time. We can now start dating these mutations of when they happened. So for example, these 6 mutations right here in this block of equivalence represents about 18 generations. The important thing I want to stress here is that each of these lines represents a distinct human being. And if we had the information, like I said earlier, we can translate each of these lines into personal names. One example of a line on this tree representing a specific person is mutation FT33300, which represents a family clade. So family clade is a haplogroup within genealogical time. And it points out here, it's pointed here in the block tree. This mutation was first brought into the world by the birth of a man named Joseph Juan Gualberto Martin, who was born in 1767. He was born in Las Trampas and as a young, newly married man, a first settler of Llano Largo, New Mexico, around 1796. So before his birth, this variant, this mutation, 33300 had never existed in humankind before. No one on the planet had that one change in that one location on their DNA happen. And until he started having sons, that mutation was completely unique to him. So, he's walking around early 18th century, Llano Largo, and he's the only one that has it, right? No one else is looking around, right? So in FTDNA terms, he had a private variant or a private mutation. So if you look here at these turquoise blocks, these are private variants. These have not been named. That means there's a person walking around right now who was born that until this person has sons, it's just private and therefore it's not going to show up on the tree. Now, it doesn't mean that now in this instance, for example, in this instance, this person is me. We'll get into that later. I have sons, I have grandsons. It's just they haven't taken the test yet. As soon as my son were to take the test or a grandson, this would split and we would have a new branching point. And I would then my line would then branch out from my brothers and my uncle and my first and second and third patrilineal cousins. And that's the point, is that same concept goes back in time. So that, 200 years from now, somebody might want to know, do you descend from Lee or Alex or Daniel? Well, I don't know. We've lost the records. Well, we can find out which of the sons this guy or grandsons or great grandsons of my dad by doing a DNA test. And so this signals that we're trying to find out in the past is still going on today. We're manifesting them for our descendants today. All right. And I guess the point again is this, is that because FT33300 was private to him, none of this stuff underneath here existed until he started having sons. And then boom, all of a sudden you have things happen. See what I mean? All right. So I'm going to stop again and see, and I'm going to take a look at the questions Q&A section here. All right, I'm female. My mom took a DNA test. My dad passed away, but his sister took a test. I'm going to take a DNA test. Should I have my brother take a y-DNA test too? Will I get better information from his test since he's my closest male relative? So the answer is yes. So your mom took the DNA test. She took an autosomal DNA test, or maybe a mitochondrial DNA test, but your mom couldn't take the y-DNA test, so you don't have one for your mom. You wouldn't. Your dad passed away, but his sister took a test. But his sister wouldn't have the y-DNA. She didn't inherit it. Only her brother would have inherited it. So that doesn't work. So I would ask the question, so if, let's say your dad's last name is Garcia, do you have a Garcia cousin that descends from the same line as your dad? And I would then have him take the DNA test. And then yes, you'll have information that none of these other tests can give you, because now you know about that patrilineal Garcia line. And you may find, oh, look at my matches. They're all Garcias, and this one Garcia that's a match has amazing genealogy, and I know that genealogy is my genealogy now. Or you may find there's not a single Garcia on any. I have 30 matches and not a single Garcia. Maybe I'm not going to go too far back before the Garcia isn't really my surname. So you don't know until you find it. But absolutely, based on what you told me here, you would definitely benefit from an uncle or a cousin, male Garcia, taking that test. The next question, I don't have any males left in my family. How can I make use of this information? Ooh, that's a rough one. So let me tell you about my Archuleta line. I actually had to recruit a fifth cousin. First of all, you know how hard it is to find a patrilineal first cousin? I mean, you have to have a pretty robust genealogy to even identify that person. My grandfather had only daughters. He passed away before there was DNA tests. There are no males left. I actually identified, and this is a cautionary tale, I actually found a relatively close, like a second cousin. I'm like, oh, I need to get this guy's DNA. And it was hard for all kinds of reasons I won't get into. On the day that the day after I finally made contact with his family and got them to agree to meet with me to talk about this, he died. Yeah, so then I'm left with this fifth cousin, and fifth cousin's far. I really would like to find, so I'm still searching for that closer Archuleta line male. I mean, it's just really hard to find sometimes. On the other hand, on my Martinez line, I was able to identify a whole branch of third cousins I never knew existed that, we met them through because of this test. And then when we went to their house, there are pictures of my great-grandfather in their house that we'd never seen before. I mean, it was just amazing, right? So everyone's different. It all depends, but it's hard. If you don't have a male, you have a male in your family. They're walking around. You just don't know who they are yet. They could be a fifth cousin. You might have to go back 200 years, but they're there. So that's where I refer you back to traditional genealogy. You have to go back to the traditional genealogy to get the data so that you can now go and get yourself more involved. And sometimes that may be years of going back to traditional genealogy until you have enough information to come back and leverage genetic genealogy. Next question, what is a private variant as seen on the block tree? So that private variant, we're going to get into that more detail here in a minute. So I'll answer that. But the bottom line is, so when I was born, I had a mutation that my dad didn't my dad did not have. My brothers, and therefore my brothers didn't have, and my uncle, my first, second, third male cousins, none of them have this mutation. I'm the only Martinez walking around with the mutation. Well, I was until my son was born. And then there were two of us. And now he has two sons. So now there's four of us. And that's how it works. Now this private variant, if my son or my grandson were to take the test, it would get named, placed on the tree, a branching point, and a new family tree, and a new family clade, 55-year-old family clade. So I'm going to get on my soapbox here. I've been saying this for years. We need to get away from talking about thousands of year old haplogroups. That's so 3 years ago. Haplogroups can be as young as you are. You can, and that's why let's move away from haplogroups towards family clades, which are haplogroups in genealogical time. Let's talk about that. So otherwise, you can say I'm R dash whatever, and like, okay, so are 20 other million people in Europe. So that doesn't tell me anything. You really need to get down to that level. And these private variants are awesome. Remember, the private variants happen on average every third generation. So it's not like I'm like some weird, well, I am a mutant, right? Technically, I'm a mutant. I want to join the X-Men. But yeah, so I have a private variant. And I'm the lucky lotto winner, and every third generation or so it happens, right? And I'm just happen to be that person. Therefore, I'm creating a branching point that will, in hundreds of years from now, may be helpful for somebody. Next question. This I just had my brother upgrade from 111 to Big Y. That's awesome. Remember, technically not an upgrade, meaning which hopefully will make you feel better because you spent a lot of money on that Big Y, and they spent a lot of money on that Big Y because they it wasn't like they're now taking that money and putting it in their pocket and giving you information they've been holding from you. When you got that upgrade, they actually had to rerun your test from scratch. So it's a completely different test. So that's awesome. So besides having my brother take the test, I also need another Garcia male that's related to my father, like a nephew. Yes, if you're a female and you have your brother that took the test, you want somebody close to your brother, like another brother or a father or a nephew. That would be great. But that's it. Then you stop. You don't take any more tests like that. The next tests you're going to be taking are going to be fourth cousins or eighth cousins. You only stick to 2. Any more than 2, now you're just wasting money because you're not going to get any more information. And the answer is, why do I have a close relative take the test? You do that to name those private variants and create a branching point. All right, so back to the presentation. I'm glad I put these little questions breaks in here because there's lots of, all right, so I'm going to go just to show you how things are quickly moving. These slides are now obsolete, and I created these slides just weeks ago, and now they're old and they don't even look like this anymore. So that's how quickly things are moving. So I'm going to move to a real live version. I can find it. All right. Yeah, so I'm going to go here. Yeah. No, I'm going to go this way. Okay, so if I hadn't purchased a Big Y, and I just, but I'm curious, I would come to this website and I would enter the information here. And it would show me information, but not my matches and stuff like that. I could also go through here or here or here. I'm not sure why they had to put it all over the place. Like, I get it. All right. So I'll come here, and there's nothing on here that's proprietary or anything like that. So, so this is the kind of information that you get. And so these are the tools I want to talk about. This is the Discover More environment. And you have country frequency, notable connections, migration map, ancient. So I'm going to blow through these really fast. But I'm going to, and then we'll come back to them if we have time later. So here's your haplogroup story. So this is basically tells me, this Lee, this is your haplogroup. This is your family clade. Represents a man who was born around 100 years ago. So, right? Now remember that technically there's another branch underneath this, and there's another branching point. It's just that because my son hasn't taken the test, the branching point isn't shown here. You don't see it. The world doesn't see it, nor will you, because my son hasn't taken a test yet. Back to the question of, should I have a brother or uncle take the test in addition to my, and yeah. Although I have a little trick that I'm going to show you here in a minute if you don't want to spend the money. Like, I don't want to spend the money for that. I want to have more grandsons. My grandsons are little. Before I want my line to be more established before I start putting it on the tree for everyone to see. But it basically tells me, yeah, this guy was born around 100 years ago, and it corresponds with 95% probability he was born between these dates. And it branched from this line. So I'm going to go to that line because I know this person too. So I knew who FT5172 is. That's my great grandfather. But I also, it's a subclade of this family tree, this haplogroup, which I also know who this is. And so you can see that this person was born around 150 years ago with a big range here. And so forth. Hang on. All right, so I'm going to go more live. So I'm going to go to the scientific details for, and this is So for those of you who've taken statistics, this will be easy. For those of you who haven't, I'm going to try to explain this really simple. So think about a bell curve. Well, this is a bell curve. It's not a perfect bell curve, but it's a curve here. And so what we have, let me give you some examples. So let's use IQ. So IQ fits on a bell curve. And so the average, the mean, and the median and the mode, the line of central tendency is 100. Okay, an IQ of 100. If you had an IQ of 100, you're the epitome of average. And then you have what's called a standard deviation. So one standard deviation is 15. That means 85 to 115. If you're in a population of people and your IQ is between 85 and 115, you're one standard deviation from the mean. And yeah, most people are going to be one standard deviation from the mean. If you go 2 standard deviations from the mean, now you're at like 70 IQ to 130 IQ. Well, what percentage of the population is that? Well, that's almost everybody. You've captured almost everybody. If you go to 3 standard deviations from the mean, now you have the genius IQ out here with Einstein, and then you have someone that maybe needs to live in a group home over here because they can't live independently, and you've captured everybody. So that's the concept of a bell curve. And so that's what you have here. You have 1 standard deviation from the mean, 2 standard deviations from the mean. And so that's why the probabilities get bigger, right? There's a 95% chance this person was born between this really long line here, and so forth. There's a 68% chance they were born in here. And so you may see in articles coming out when they're trying to guess or estimate when a mutation occurred, they'll use this data. And so this is really important because then it tells us, and I'm going to kind of demo this here for you here in a second. All right. So suppose I had no genealogical data for my line. Let's say I was adopted and didn't know anything about my patrilineal line at all. The Big Y700 could still identify the mutation of my patrilineal ancestor who lived around 150 years ago, right? That is shared by others. So given this hypothetical position, this hypothetical situation, no genealogy, it would be a good idea for me to reach out to my identified Y-DNA matches who also inherited this variant, as we share a common patrilineal ancestor within a defined range within genealogical time. So again, for the more data we have, and or the farther back in time we go, the more accurate the prediction. So this range, right, tightens up when you take the more tests. So they're like, why are they asking me to take more tests? Don't we have enough people testing in this project? The more folks that you have testing that we're strategically selecting, that curve that gets tighter, and therefore the time estimates become more exact, if that makes sense. So I'm going to give you an example. So here's the genealogy for that person. This is Joseph Juan Gualberto Martin, otherwise known as haplogroup FT33300. This is him. He was born with this mutation, right? So here's his information. I know when he's born in 1767. Here is he's the ancestor of all the Lobatos and Martinezes of Llano Largo, New Mexico. And these 2 family lines, identified in blue and yellow respectively, are family clade FT33300 and FT52172, which is a subclade of this one, right? So this one occurred in Joseph Juan Gualberto Martin, which then got passed to all of his patrilineal descendants and will forever. So all the men that descend from him continue, like I said, I have 2 grandsons. They have this mutation. 1,000 years from now, if I have my patrilineal line were to exist, not daughter route as they call it, this mutation would still be found in that line. So this phenomenon is additive, which means that I have this and this, and then it keeps just going up backwards in time, all the way back to Y-DNA atoms, which is kind of cool concept. So what we have here is a branching point. So we have this guy, he's born in Las Trampas. He, as an adult, he's a first settler of Llano Largo. And it's always awesome when you have somebody who then moves away, in this case, up the mountain, and then has children, because then now you're physically in a new place with a new mutation. He has 2 sons. Now, it's interesting how closely these families are. You can tell that these folks, if you're familiar with Llano Largo, right? It's a farming community. I can just tell these folks all live next. These are abutting farms. You can just tell, right? And let's see how, for example, they're intermarrying each other. So I'll be honest. What probably happened here was Mr. Lobato went out to the field, and Jose paid Mrs. Lobato a visit, and Mr. Lobato didn't know about it. And this is why I'm talking about where Y-DNA is more accurate than a birth certificate, because when this guy's born, I've seen this genealogy, right? The Lobato line keeps going. That genealogy is not right. I know for a fact that this person's not the son of Mr. Lobato. I know that. You can run, but you cannot hide. You can die thinking you took your secret with you. 100 years later, we're talking about it, right? These are brothers, and what happens is he then has a son, and he has a son. So these are first cousins, and then he has a son, and he has a son, and these are second cousins. And then he marries his second cousin once removed. So this person's walking around not realizing that this is actually Martinez. It's the same line. But this is common. There's nothing, right? This is, if you lived in Llano Largo, marrying your second cousin once removed is actually the rule, right? And here's an example. Cayetano has a son, and Mr. Madrid has a son, and then, you know, he has a son and he has a daughter, and they marry each other. And then these 2 folks here, I'm sorry. Yeah, are siblings, right? Actually, I'm not showing it here. Oh, here it is. I'm sorry, wrong line. Here it is. Sorry. These are the 2. This is my grandfather, my paternal grandfather, and this is my grandmother. And so if you look at this gentleman right here, he has 2 sons, right? Jose Ignacio and Maximo. He has a daughter, and he has a daughter, and these are first cousins. And then she has a son, and she has her. And so he ends up marrying his cousin, right? So this is normal, right? But the fact is that these are all descendants. This is me right here. We're all descendants of this one guy. I have this family clade. I actually have my own now family clade that's not on here because I haven't tested yet, but I know I have a mutation. We'll get to that in a minute. So I have a family clade, which is a subclade of this family clade, which is a subclade of that family clade, and then it just goes back. All right. So back to the statistics. So this is, instead of just putting FT33, we can put his name. And then here was the guesstimate, right? Here was the statistical estimate, and here's the actual. See what I mean? That's pretty darn close. It's not exact, right? But it basically said, hey, there's a, 95% chance this guy's born between this date and this date, and sure enough, here's the date. So now this is all based on just that small data set. So I go back, how many testers, big Y testers? 1, 2, 3. So based on 3 big Y tests, I'm able to get this close to the actual date. If you have like 20 tests, if you're a part of a bigger group, a bigger project, you have your date is going to be closer to the estimate, to the mean, right? So I'll show you another example. So let's go up a generation to BY 93730. So this person had this is Nicolas de Espinosa. You guys probably know all about Nicolas de Espinosa. He's the father of all of these folks, right? Including FT33300. Okay. So that means that we now have more testers. We have all these Espinosa people, lots of people who don't have the Espinosa name. And so now we've doubled the number of testers. What is doubling the number of testers going to do for our data? If we come here and we look at, so according to the calculation based on SNP and STIR test results from 7 testers now instead of just 3, there's a 95% chance this person was born between 1593 and 1830. So Nicolas de Espinosa was born in 1673. So you can see that I'm getting closer to the mean because I have more testers. Does that make sense? So now this is all great, but what this really helps is when you don't have that birth certificate and you're trying to figure out when was this guy born, right? This is how this tool can help do that. Again, the farther back you go and or the more test data you have, the closer the age estimate will get to the mean and the more accurate it will be. So branching points play a particularly useful role in the analysis of the big Y block tree, especially if you can identify who the person is via genealogy. Now, there's another person I can identify, like I mentioned, on this tree who had a private variant, and that's me. So here's me, right? I have a private variant that hadn't existed before, right? And here's the trick. For those of you, if you are like me and you've taken the test, the big Y test, and you're sitting with one private variant, here's another person, another example right here, right? If you find yourself in this situation where there's no such thing as average of one, this is one person. So it's not really an average. This is an average. There's an average of 2 private variants with this group of who knows how many people, right? But here, if you have an average of 1, you're really just one test away from there's a branching point coming. I didn't want to have to spend the money. So what I did is I went to the private variants part of my DNA results, found the DNA. This is the address. This is the place where this mutation happened on this location. I went over to the International Society of Genetic Genealogy site, entered in that information, and bam, there's my family clade. So I know that if my son or grandson were to take the test, the new branch will be called FT4174. So I know what my family clade is, even though it's private. It doesn't, and it's not out there to the world. Actually, it's in a database somewhere, and I was able to go find it in the database. So we now have a new branching point, FT4174. That's me, right? And so my son and my grandsons have this mutation and this mutation and this mutation and so forth. So it's additive, right? They have now a new mutation that my brother and his son don't have. Me and my kids and my brother and his son, we all shared this mutation. But his line and my uncle and my other brothers and my first, second, and third cousin Martinez's don't have this mutation. And therefore, forevermore, for as long as there are males in my line, this will always identify me as an ancestor versus others. And the point of this is that this works backwards in time to solve the same kind of, if someone wanted to know, do you descend from Lee or do you descend from Alex? 100, 200 years from now, they want to know that question because somehow all our great records got lost. They could take a big Y test and find out, oh, I have this marker, you don't. I descend from Lee, not from Alex. And that's how we solve these questions. And you're going to see, again, in these journal articles where they're saying, hey, we can start identifying which line you came from by using the same concept. All right. All right, so every man on the planet carries a Y chromosome, which was carried by his biological father and that father's biological father and his biological father and so on. And it's passed down. As it's passed down, a record of all previous mutations gets passed along as well. So every man carries the full mutation history from themselves back to Adam. See what I'm saying? So now my son, now I have one more mutation than we had before. And then if you go back in time, that number of mutations is going to get smaller and smaller and smaller until it gets and coalesces around this guy that lived 200,000 years ago that we call Y Adam. So if I wanted to navigate back in time, I can go to, hang on, where's my block tree? All right, that's timed out. So I'm going to go back into it. What I'm going to do is I'm going to go into my block tree and I'm going to show you how I can go back all the way back to Y Adam. And the point is that this concept goes back and forth in time. And while that's thinking, I'm going to look at the questions while let's see. My male relative had WGS done and we can obtain the raw data. Can it be analyzed instead of purchasing Big Y? Well, the problem is that does WGS give you your matches? And if you don't have matches, then I don't know how helpful it would be. So that's my answer to that. That's the point about Big Y, is that it's not just the data. The data doesn't exist in isolation. The data is only helpful if you can identify other people who match you. And that's what family tree DNA is giving you. All right. It logged me out. Next question. Did you say that a haplogroup can be as recent as this generation and not merely centuries ago? Yeah. So I just identified my family clade, which is 50. I just had a birthday. My family clade is 55 years old because I'm the father of my family clade. See what I mean? If so, a haplogroup can simply occur, as you mentioned, above your own paternal line. Yeah. So I have 3 family clades. In other words, I have 3 haplogroups within my within genealogical time. Next question, do you make the block tree yourself or is that generated with a tool? Yeah, it's generated like this. I click on it and it just appears. Now I won't show you the bottom of it because that shows my matches and that's private, so I'm not going to do that, but I'll show you the top part, which anybody can get to. All right. All right, so this is me. This is my 3rd great grandfather, 5th great grandfather, Nicholas Tespinosa, and then you just keep going back in time. And if you go back in time, this is a subclade of this, which is a subclade of this, which is a subclade of this, and so forth to, oh, there's QM3, right? So let's look at QM3. QM3 is basically means that 15,000 years ago, one of the men that crossed the land bridge into the Americas that founded the Clovis culture, that's QM3. And the vast majority of men in South America and Mesoamerica are QM3. And then lots, maybe up to 60% of them through Canada. And so that's how, I went back in time. And I can keep going though. I can keep going back. And let's keep going back before Q. And now I'm P. And let's go farther back in time. And farther back in time. Hundreds of thousands of years. Or I'm sorry, tens of thousands of years. And I just keep going. Because I'm not Q anymore. Q hasn't even been born yet, and now I'm in P, and now P hasn't been born yet. I'm into K, and I'm just keep going, right? K haplogroup, and maybe I'm at 30,000 years at some point, and I just keep going until I hit Y DNA, and all men will end up at the same spot at some point in time. All right, so I'm gonna bring. Let's see. All right. All right, so in review, SNP is a single base pair substitution occurring in the Y chromosome, and each creates a mutation, which creates a mutation, and sometimes identifying a branching point, which is what we have here. There's a branching point between me and my siblings. I have lots of brothers, but this is just one of them, right? So my line is now branched from my brother's line because of mutation happened, one on a specific line of code. So here's a big one, and this is, I'm not going to get into it because there's I believe there's a paper coming out in December on this topic. But you saw how accurate the time the most recent common ancestor statistic was based on just 7 testers. Imagine how much more on point these data estimates will be for a much larger project. Let's say one 4 times larger, like 27 testers like this one. So Darius Celeste Landris and Miguel Torres will be authoring a paper on the big Y block tree soon. And their presentation will be based on strong statistical data and will be very interesting. So my point is this: when you're reading that paper, you see the estimated dates they're giving you, now you know where those dates came from. And because of the sample size you see here, that those numbers are going to be super close and super accurate. This is my Trujillo line, my one line that actually goes back to a progenitor. And so here you can see this is this represents Cristobal El Viejo Trujillo. And so you have branching points. Most of the folks that took the test who have the Trujillo surname fall under this right here. They need more testers. They have this old, this old haplogroup, which I'm not a fan of, and other folks, these that don't have the Trujillo surname, have taken the test, and they now know more specifically how they descend patrilineally from Cristobal Trujillo. So this one right here, the ancestors Tomas Sandoval, born in 1714. So Tomas Sandoval is a direct patrilineal descendant of Cristobal Trujillo. Then you have the Penrod surname, which occurs around 1890, and that surname before Penrod, at some point, if you go back in time, is a Trujillo. Same thing with this one, and so forth. So these branching points are super important. All right, so I'm going to see how we're doing with questions, and let's do a time check. How much time do we have left? It's 20 after 10.
It's 20 after 11 on our time
zone.
Yeah, so you've been at it for an hour and 20 minutes.
Yep. So I'm going to stop here. I could keep going, but I'll stop because I have slides on slides on slides. But I want to make sure. So I've listed. So this guy, I put the little family clade on this grandson, right? Kind of cool. So I want to open it up to questions. If somebody wants to, for example, look, have me look at their. I can just throw out a family clade at me, and we can look at the data if you want, or maybe we're out of time. We can do whatever you want.
Lee, can I ask you a question? This is Henrietta.
Yep.
So because you keep mentioning that the big Y, and I mean all of the DNA is moving relatively quickly, when we do something with the charting or the collaboration that goes on with this, I keep saying we need to put a date with it because next week it'll change.
Yep.
And then you'll be in this infinitesimal argument with somebody because they're looking at some data that you're not looking at based on a date, right?
Exactly. Mm-hmm.
Okay. So, and then the other part is also the paper trail, the old-fashioned genealogy. Our program, we actually help validate the lineages. We check each other's work, and I can't say enough in terms of what you're talking about, trying to find like that third or fourth cousin to expand your genealogy horizontally, not just going straight back. And I think that's kind of what you were saying about some of these ancestors that you've had to go hunt down, right? Is that you've expanded the cousins and the brothers and everybody, not just going up your Martine line and not doing anything else.
Exactly. So like I said, I've done I've done the 4 lines, and if I want to, I could go back and do all 8 lines and 16 lines and so forth. This is the Martine Serrano line. Oh, I didn't show you the time tree. Let me show you this really quick. This just came out Thursday. So if I can find this person, here's the Martine Serrano family. And so here we have, so now look, so this is more advanced than the big Y. So the big Y block tree feeds this. So this is the family clade. I'm sorry, this is not a family clade. This is the haplogroup for the Martine Serrano family. And these are all the lines that have come from that family so far. And then you can see that they're putting dates now. And so, yeah, you can actually look at this and date when that mutation happened. And because this is such a large group, these dates are going to be really on point. Does that make sense?
Mhm.
So that's amazing. And then it gives you the range, right? So it's going to give you it's like this person was born in this date, but 65% chance or 95% chance they're born sometime within this time frame here. So these are the tools. And again, what I'm showing you here, anybody can get to. You don't have to be purchased a big Y test. You don't have to have any affiliation with family tree DNA whatsoever. And you simply go in there, and then you have access to all these tools. Kind of cool stuff. There's like notable connections. These are people that have, you have a paternal ancestor 4,000 years ago. You and George W. Bush had a paternal ancestor in common. So there's some gee whiz stuff here. Here's the migration map for the Martin-Sedano line over time. And it shows, so Y DNA atom, right? And then it shows you how that Martin-Sedano line. Now, the thing is that it doesn't have the connection to the Americas yet, which they really need to get that fixed.
Okay, you
Yeah,
have some number sequencing in the chat feed, so you need to go in and start typing those
oh, okay.
in.
Yeah. All right, so let's start at the top here. And again, you guys can go in here and play with it this morning. You can go in here and just go into the website 27. All right, so this is the haplogroup. They were born around 250 years ago, plus or minus 150. And then let's look at the time tree for that person. So this is a Q, so this is like a QM3. And so you're going to find there's not a lot of data, but then this gives you kind of the estimated date of when this actually, this is a family clade. It's within genealogical time. And so there's let's do another one. All right. This person was born around 150 years ago. So again, these are family clades, right? These are, I love these. These are within genealogical time. If I come here, find the person who's in red, because that will be you. Where are you? There you are. Let's click on that. Oh, let's go. That was better. Trying to get the right view. So it looks like you have somebody that came, keep going back a little bit farther. All right, so that went back. So let's see. I went back a little bit too far. So for me, I wouldn't, anything before like 1500, I wouldn't really, you're now outside of genealogical time. I would stay in here and kind of take a look. But so you have lots of family clades, right? Cool, good stuff. Actually, let me look at that one more time. Let's look at their migration path through time. Do that really fast since they're from the other. There we go, migration map for that one. So this is the migration map for, oh yeah, they went up there. There's a way to make this bigger. Maybe it'll let me. You'll have to play around with this, but it's showing the migration path all the way through and into the United Kingdom. It's interesting. Let's take a look at another one.
We also have a question from Anna. She said, "Is the big Y the same as the big Y-700?"
So there's the big Y 500. There's the big Y, and then there was the big Y 500, and then there's the big Y 700. So the big Y, if it's just big Y, it's an old test. So if you were to take the big Y 700 as an upgrade, you might get more information than you currently have. Now I'm skipping some of the haplogroups are sending me because they're too old. If you send me a 10,000-year-old haplogroup, then I'm probably not going to go there. You need to take a big Y test and get something a little bit more recent. But this is
so like the JM172.
Yeah,
that's a common.
Yeah, now this one's still pretty old. You know, this is still, we're talking about 1600 years ago. So whoever this was right here, you need to take more testing, because if you take a test for yourself and then a close relative, you're going to get a family clade. Let's do this J. I have a J. My true line, I think, is a J. See here. This is, again, pretty old, 3,450 years ago. So if you have a haplogroup that's old, like this old, you're a candidate for a big Y test. You want to take a big Y test. If you've taken a big Y test and you still have a haplogroup that's old, watch my video on the rule of 4. It shows you how to build a family clade. And that's what we're trying to do. We're trying to build a genetic profile within genealogical time, so we can start using all the paper that we've been working on for decades of time together with the genetics to start solving genealogy questions. Right before, and this was the argument about not doing a big Y 5 years ago, 3 years ago. It was like, oh, what's a 1,000 or 2,000-year-old haplogroup going to help me with genealogy? Good point. But that's so much 2 years ago. Right now, like I said, you can, if you know the right people to test, there's no reason why you couldn't have. Not everyone's going to have a 55-year-old family clade, because maybe you don't have a mutation. Maybe the last mutation for you was 200 years ago. Remember, I said on average every 84 years. Well, that's an average. We have some lines that 200 years have passed and no mutations, and that it happens, right? But you definitely should be able to find a haplogroup within, the time frame of the history that we're talking about in New Mexico, right? 1598, 1680, 1695, those kind of dates, right? There's no reason why you can't start to have haplogroups, family clades that we call them, within those time frames. And then we can start having a conversation, and then we can start talking and writing in these journals about people we know and lines we know, and identify them and say, here's this ancestor, here's their family clade. And if you ever take the test in the future and you have this family clade, guess what? You now get to piggyback on all of my stuff. And you don't even need to connect with me through paper, because now you've connected with me through through genetics. Now you can start the paper and you can start working your paperwork towards me, but at least now you have a direction that you're going.
Okay, you have one more question from Maria. The big Y 700 cannot be done with the DNA from a previous Y36 test, correct?
Yes and no. So I wanted to upgrade my my Trujillo line from Y37 to a big Y. And when I sent it in, I was hoping they could use the same sample. Unfortunately, the sample was 7 years old, and they're like, nope, you have to get another sample. So I drove from Gilbert, Arizona to Socorro, New Mexico, and back, so I could manually get a test from this elderly nephew of my grandmother's. Yes, I'm dedicated. I had to do it again. So if you've taken a test within the last 2 or 3 years, you probably don't need a new sample. They can use the same sample as the Y37. But if it's been older than that, they're going to send you they'll send you a new kit and say, we need another sample. Hopefully that answers that.
Okay. And then Laura, you have the JY130866 up on the screen. And her question is, this is my brother's big Y700. I have him in several projects and have not found any matches. The line has dotted out. Any suggestions?
Yeah, this is the same situation with, like I said, with my Archuleta line. This is the only thing you can now the answer to your question is no longer in genetics, it's in traditional genealogy, right? You're going to have to go back. And so if the line's been dotted out and you're like adopted or something, you don't know, or you just don't have the genealogy, you're in a bad place, because you don't have the information to proceed. But the answer is going to be in traditional genealogy. You need to find a 4th cousin or a 5th cousin. You need to find somewhere there's a male walking around that has that DNA. Even if 3 lines back have dotted out, the 4th line back, right? There's somebody there. So it's hard, because it's not hard. It's not easy to find a 4th patrilineal cousin. That's a very specific thing to look for.
Okay, so I think Suzanne's question's the same thing. His sample's from 2005.
Yeah, it's way too old.
That's way too old.
Yeah, and so if he's passed, you can ask him, like you can say, you know what, there's no one else that can get the sample. This is all we got. I'm willing to pay the price, understanding that you're not going to be able to get a good, and then I've spent all that money, but I want you to try anyway. And they may or may not do that. I don't know. Kind of puts them in a weird position because you're asking them to take just for your money to go towards a test that they're like, I can't guarantee we're going to be able to get a good sample of data from this. But if it's it's either that or we have to dig up a grave, that who knows, they may, I don't know. You might want to reach out to them and ask them if they're willing to do that.
Okay, so we have another question from Ron. How would you match my Y-DNA with ancestors if my paternal line father and grandfather and siblings are deceased?
So your father and grandfather, so if you have a great grandfather, those would be what, your third cousins? Do you have third cousins with your paternal family name walking around? That's what I did. I had third cousins that I identified take the test. And then I found a fifth cousin, patrilineal fifth cousin, that Lobatos was a fifth cousin to take the test. But again, you have to have a really good genealogy to even know who those folks are, right?
Right.
Now, yeah.
And the other part is just to wait, because like if it's a Y-DNA and he's a Pinot, there, you just need to go find a lot of Pinots to test.
Yeah.
They might just want to do a 67 marker test, and then you can kind of go from there.
Yeah, as a matter of fact, if I was shotgun it, I would do a Y37 test because those are the cheapest ones. And the purpose of that is just to find, in the old days, you do a Y12, right? But now those aren't even available anymore. So I would do the cheapest test. I'd shotgun approach and spend, it's not that expensive to do a Y37, and then cherry pick from that. If there's something comes up and says, okay, you have what I want, I'm going to now invest in the Y700 for that one line based on what I see from the Y37. That's one way of doing it.
That's a great idea. Okay, well, we're going to end today's program. I think Lee's worn out. And I just want to, again, on behalf of NMGS, the board, and all of our members, thank you for your lecture today. I'm sure it'll be informative. We will be putting it up on our YouTube channel. And yes, Daria and Miguel have written a Big Y article for the December journal that will be out late November, early December. And we look forward to seeing that. So Lee, again, muchas gracias. Have a great weekend. And we'll see you around. Say hi to your mom and dad.
I will.
All righty. Thank you, everyone.
Hey, everybody.
Bye-bye.